Eastern branch of the Entomological Society's president, as well as a gazillion other committees. And he's also served on the governing board for the Entomological Society of America. And during that 6-year term, he was able to help formulate and influence the direction that our professional society has gone to. So he has 87 publications to his name, I believe. Or at least that's what it was.
Research-wise.
Research publications. I'm not counting the extension publications because nobody can count that high. So with that, George, please.
Okay.
All right, give us a list.
Well, thank you, Ann, for that introduction. You took away some of the things I was going to talk about at the beginning, but that's okay. That's not a problem. I'll go over it again. For those of you that have never met Dr. Maramorosch, this is Carl down in the my left corner, okay? He had a very illustrious career, and he was indeed 101 years old when he passed away. He was actually on vacation over in Europe at the time, and one of the many things he liked to do was travel all over the world. Our current group of secretaries, I don't ever think saw this, but Carl was also an accomplished magician, and he used to do tricks all the times for the secretaries. And he was also an accomplished pianist, okay? So he well-rounded individual.
And spoke like 25 languages.
Oh, and spoke 25 languages,
What?
yeah, absolutely.
Like literally 25?
Okay,
Yeah,
why
well,
are we
maybe not
moving?
25,
Okay,
maybe I'm under-
so Carl was born in Vienna, and he grew up in post-World War I Poland, okay? And when World War II hit, he was able to escape from Poland and ended up in Sweden. And then in 1947, he immigrated to the U.S. And he received his PhD from Columbia in 1949. He went from there to Rockefeller University, where he had a very illustrious career. He joined the Waxman Institute, which is here at Rutgers over on the Bush campus, in 1974 as a distinguished professor. And in the mid-'80s, he came to our department, and as the story goes, they kicked him out, and he didn't want to quit. And so I'm not quite sure how he got connected with our current chair at the time, which was Don Kakamies, but all of a sudden he had an office in our building, and he remained with our department until he passed away. And so he was amazing. He used to drive from Westchester County in New York.
That's where I'm from.
Good, good. And down here on Mondays, he'd spend Tuesday, Wednesday, and Thursday here, and then drive back up to Westchester County for the weekend.
So he was still driving at the age of 98.
I'm sorry? He was still driving at the age of 98. At that point, he had to stop because, and he actually had to have somebody, assisted care person, taking care of it, which went with him on his vacations. So his expertise was in invertebrate pathology, basically looking at the relationships between insects and plants. He did a lot of work with leafhoppers and the diseases that they transmit. Ann said, I have 87 publications. That comes nowhere near Carl. He had over 800 research publications, including articles. Well, he had more time. I'm not going to make it to 101. Yeah, including papers in science. Published over 100 books. Most of those books, I think, were probably written once he joined our department. And he was always working on some compilation of, specifically in plant pathology. He's a winner of the Wolf Prize in 1980, which, if you're not familiar with that, that is like the Nobel Prize in agriculture. And he was also named in '83, before they threw him out of the Waxman Institute, the Richie Starkley Professor of Microbiology here at Rutgers. Okay,
What year?
1983. Okay, so this series was begun in the late 1990s. It was the idea of Don Caccamise that we should be honoring Carl because of his career. And the decision was made by the department to begin bringing in graduated or alumni of our graduate program. And so we did do that. Dr. Sunny Ramaswamy was the first person invited in. He was a physiologist, I believe, of sorts. And he, at the time, was chair of Louisiana State University's entomology department. He then moved to Kansas State as their head, went to Purdue as their director of their experiment station, Oregon, and became their dean of agriculture there. And then probably the most impressive thing that Dr. Ramaswamy's done in his career, President Obama asked him to be the director of USDA NIFA, which is the organization that a lot of us receive our grant funding from. And he did that up through the first year of the Trump administration. Now, as I said, Carl passed away in 2016, and so in that year we changed the name, of course, to being the Memorial Lecture Series. Okay, so your speaker today.
Some picture, George.
I didn't think I was going to get laughter with that picture.
It's a good laugh.
I just want to point out that this is something that when I came was professionally done because I was an extension specialist. We used to have in J.B. Smith Hall, which is now the Community Gardens over at the center, a communications group. One of the members, in fact, the person who ran it, was a photographer, a professional photographer. And every new faculty member that came in at that time, I was told to put on a coat and tie and come down and see him. And he took this picture because at that time, when we went to county meetings and state organization meetings, they always wanted a picture of the speakers. And so this is much better than I would ever be able to take of myself. And we couldn't do selfies back then. So, yeah.
It was kind of difficult.
Right.
Bigger size kid.
Yeah, right. And, you know, I have changed. My hair's a little longer right now, and it's a lot grayer. But anyway, as Ann said, I came here in 1981 to work with Dr. Fred Swift, who was our tree fruit entomology. And I did a lot of work with him, and I did my research involved the green apple aphid, which is a pest of apple trees here in the state. Again, as Ann said, I got my PhD in 1985, and in 1985, I'm not sure it was the same day, but when I graduated, Fred went on a sabbatical, and the university hired me as a postdoc to take care of his research program while he was on sabbatical, and to also teach ag entomology and pest management for the first time. I had never done that before. I had never taught a university class. I had never been a TA. Rude awakening. Yeah, so, and they didn't give me a lot of time to put it together, so Dr. Hawkins, I know what you went through when you came here. I then moved to Rutgers as not quite a professor. It was a consultant. Things worked a lot different back then. So Mark Robson, which I know several of our faculty know, was actually housed in J.B. Smith Hall. And he left Rutgers. He was in charge of our pesticide applicator training program and what was called NAPIAP, which was a pesticide impact assessment program, to go work with DEP. Went to the bad guys from the good guys. At the time, that's what they were considered. And he made a couple of phone calls, and I got invited to go over to Martin Hall to talk to the chair of the Ag Extension Specialist Department, which we used to have. And he said, well, based on Mark's recommendation, would you like to take this job? And I did. And so they hired me as a consultant because they did need to do a search to hire me as actually an extension specialist. They did that. I was lucky enough to be chosen for the position, and they hired me as a non-tenure track assistant extension specialist in '87. And then as you know, again, things were much different back then. Extension had a lot of money then. They have almost nothing now. And I got a phone call, and so did several other specialists, non-tenure track people in that department, and said, George, we've got some money. Would you like to switch to a tenure track position? I said yes, and the next day I was in a tenure track position. Interesting situation though. I was given no lab, and so that was a problem. But at the time I didn't worry about it, because when they hired me, the search committee, the thing they were most worried about was that I would do research and not the extension that they wanted done, and it was different back then. And I had to say, no, I will not do any research. And so I didn't until I
So that's
became
why
a
they didn't give you
tenured
a
track
lab,
position.
is that
I'm sorry?
No lab.
Right,
they
because
didn't want to
they didn't want me doing
Did they give you a
research.
startup?
I had no startup.
Yeah.
Yeah. That's how I learned how, with very little money, to do what Ann was able to do. And I think we lived off of $5,000 from hatch funds. We used to get a lot more hatch funds back there. Okay, so what I'm going to try to do here, that's enough about me, is here's my outline for today. I am going to start talking about a project that a couple of years after I got here, I was asked to become a part of and help with some things that needed to be done. And you can probably figure out by the pictures that it was with the Colorado potato beetle and this little tiny parasitoid wasp, Adovum butleri. And then once I'm done with that, hopefully I'll have enough time for the second part of my talk, which is to talk about what I call an opportunity of a lifetime, and that's an opportunity to work with this stink bug that nobody knew about. And at one point, my lab was the only lab in the country that was working with that. And so I'm going to talk about some of the things we did there. Most of it are things that Ann worked on while she was my PhD student. And so just a little background in case you don't know about Colorado potato beetle, Leptotynotarsa decemliniana. It is actually one of the pests we have here in the United States that is native to the Americas. You find it throughout North America and all the way down into South America. It has been introduced into Europe and Russia, and in fact Russia at one point charged us with a complaint saying that we introduced it into Ukraine so that it would wipe out all of their tomatoes and their potatoes there, which wasn't the case. But
It would
anyway.
have been too much of entomology for the CIA to figure that one out. They would be there.
Yeah, I'm not sure how the Russians figured it out. They thought we did it because we didn't do DNA analysis back then. Yeah. So this is the life cycle of the Colorado potato beetle. The adults pupate in the soil when they're, to go, and then eventually become adults. So they spend the winter as pupa. They emerge in the spring as adults. They start wandering because they don't have enough reserves to be able to work their flight muscles, so they have to go find food. And once they do that, the males, females, of course, mate. They go lay their eggs on the host plants, and you can see those yellow eggs there. They then hatch into what the growers call slugs, okay? And they go through 4 instars. That 4th instar then drops off the plant into the soil again, pupates, and the cycle starts over again. And here in New Jersey, most of the time at that point, I'm not sure with warming what it is now, but we had 2 generations per year that we had to deal with. So they feed on solanaceous plants, okay? The nightshades, basically. And this is one of their host plants. This is black nightshade. They're also very common on horse nettle, which is what this is. And I used to get a lot of calls from homeowners that say I planted potatoes in my backyard, and all of a sudden Colorado potato beetle showed up. Well, I said, well, they were there. You just didn't know you had horse nettle or black nightshades in the local area. And they also feed on, this is buffalo burr, okay? And so, they feed on a wide variety of things. In agriculture, of course, potatoes is probably the number one plant they feed on. I like to say that this is filet mignon to them. They are capable, if left uncontrolled, to completely wipe out that field of potatoes. And organic growers do have a lot of problems even today with these if they get in, because there's not a lot of things they can do other than using BT, which the variety that kills it I don't think is readily available anymore, and they're left basically with hand picking or possibly the use of pyrethrins. They also feed on tomatoes, okay? They are reported to feed on peppers, which is also a solanaceous crop, but they really don't. I've never seen them feeding on peppers. They will go on peppers, but then they just kind of sit there. They don't do any feedings. I think there's too many nasty chemicals in the peppers for them to be able to deal with. And they feed on eggplant, which is what I'm going to talk about today. I shouldn't say I should say how a colleague of mine here in the department developed a program using that egg parasitoid to manage this without the use of insecticides, basically. And the program was very successful, and I'll just say that prior to the program being implemented, growers were spraying up to 18 times a season to control Colorado potato beetle in eggplant, and it dropped to less than 2 per year because of the program. And so this is the damage that they cause. The adults and the larval stages feed on the leaves. Adults also will feed on the fruit. And so I've kind of highlighted here the scars here, okay? They're not very big blemishes. That's from a bite from the beetle, but those bites degrade what they can get for the fruit when they harvest it, and so that's also a problem here. This is the egg parasitoid again. I apologize for the picture. This picture is very old, but believe it or not, there's nothing better on the internet out there. This is the only thing that shows up. So, yeah, and that's basically because nobody rears this anymore. And I'll talk a little bit about that if I remember at the end. So this is Dr. Jim Lasham. He retired from our department, I think probably close to 10 years ago. Okay, so he is the person that's responsible for putting together the program, and he developed a program for controlling the beetle in eggplant using this parasitoid by doing several repetitions of releases into fields. Okay, and so he did all this by starting on farms with a grower and saying to the grower, give me 1 acre. Let me show you what I can do with this insect. And most of the time they would say, okay, I'm in, can I have more, and expand to my acreage. And he had a story about a grower that, because he'd just walk out in the field with a little ice cream cup, and he would open it up in the middle of the field and let him go, and had all these little black specks in there, and they're like, they're never going to control this, you got to be kidding me. Well, it worked, and it actually became a very popular program with the growers. Of course, now it wouldn't work, and I have to give the lab credit. This is a picture of the front door of the Philippa Lampe Beneficial Insect Laboratory. They were responsible for rearing the parasitoids that he was releasing, and they're part of the Department of Agriculture. The program also included scouting, and the Department of Agriculture hired the scouts, and they managed the scouts for this program. They'd visit the farms, and Dr. LaShawn would get a weekly report of what they were finding. The growers actually paid for this. Now, by today's costs, it doesn't sound like a lot of money, but it was around $50 that they were paying per hectare to be in the program. It paid for the scouts, and it paid for the gas that the scouts were using, because they were using their own vehicles. They borrowed that from our IPM programs that we run in a number of things. That's how it works with our scouts here. And so without them, this wouldn't have worked. So in talking with Jim, we used to talk about this all the time. And we basically, at the point once it was going, there were a couple of questions, two main ones. One was, how can we reduce the number of samples that these scouts have to take, because it's taking a lot of time. And so every field that they sampled, they had 100 plants flagged throughout the field, and they would go and sample those. And then the other question, of course, which everybody wants to know, is the program cost effective? And so we decided to go and look at this. Examine the consumption rates of each life stage. They knew what it was for potatoes, but they didn't know what it was for eggplant. This will become important in answering the second question here, and that's to develop a sequential sampling program. And then the final one, of course, conduct a profit loss analysis. And so I'm going to talk a little bit about what we did to try and come up with those answers. We'll start with the consumption rate study. This was done by a couple of other people. One of them was an undergraduate. At that time, we only had a minor. I think she was working on a minor. Dr. Wong knows her, Karen Vale. She worked on this project. This was her research project. And so she would take different life stages, not the first instars, but starting with the second instars, and she'd individually put them in a box that had a divider that would come down over the midrib of an eggplant leaf that had been cut into a disc. And those discs were 63.6 centimeter discs. Basically, take a petri dish and cut everything away from it. And then she would follow their development by feeding on half of that disc until pupation. Okay, and she replaced the discs when needed so that they didn't run out of food. We did a similar thing with mated females to look at their feeding, and that was done in a 9 centimeter petri dish. And then that was run at different temperatures, because we wanted to know how much they consumed, and we also wanted to know how much temperature impacted how much they consumed. We looked at consumption by taking dry weights of material, and we compared the fed and unfed sides. And then we did a bunch of nonlinear regressions to figure that out. And so this is the regressions for the second, third, and fourth instars. And you can see, as you would expect, feeding went up as temperature went up for each of the life stages. But with larvae, the maximum temperature we used was 35 degrees C, and you can see in all cases it actually dropped. So there was a point where they actually slowed down and stopped at some point, stopped feeding. Similar results for the females. Okay, so this is the most important part as far as I'm concerned with the study. We took that data and then we looked at it, and it was obvious fourth instars actually fed more than all the other life stages. They even fed more than the adults. And so it'll become clear why, but we needed a way to go out into those fields and not have to account for the life stage and adults. And so what we did was we made fourth instar consumption 100%, and then rated all the rest of the life stages based on 100%. And that's what you're seeing here, and it was at different temperatures. And we use this data ultimately, so when we sampled, the scouts just had to walk through and just count how many individuals, didn't have to care about the life stage, okay? Which, that in itself sped up the process of having to do the sampling. So we found that consumption varied by instar and temperature. The feeding equivalence based on the 4th instars was useful, and so they plug into the sequential sampling plan that we worked on next. So if you're not familiar with the sequential sampling plan, they've been around for a long time. They were developed during World War II as a way to do a rapid assessment of quality control with things like ball bearings that were being used in the equipment. In terms of entomology, it was first used with forest insects, okay? And it soon after got adapted in a number of situations in the 70s and 80s for different IPM programs in agriculture. And to create one, you needed to be able to determine what the spatial distribution was in the field with the insects, and basically, did it fit a negative binomial distribution, a Poisson distribution, etc. And depending on which one it was more and more difficult, and negative binomial was the worst to work with. In 1975, a researcher in Japan, Dr. Awayo, came up with a method of not having to worry about that. And it involves the use of Lloyd's index of mean crowding. I don't know, Andy, you talk about that in insect ecology? Okay, good. So some of the students know what that is.
Not yet.
Although this was in the first lecture.
It was. Oh, well, I'm sorry. You've all heard this before.
It's great.
And so it took the values created by Lloyd's index of mean crowding. You did a linear regression, and you came up with the values that you would plug into the rest of the equations. And I apologize, this is going to be a slight bit of a math lecture for the next couple of slides. So Nyrop and Simmons in '84 actually showed that this method was flawed, and because of the variation between means and variances when it wasn't a linear relationship. And that was solved in '85 by Ekbom, who said we should use Taylor's power law. You talk about Taylor's too? No, okay. And so it uses the same values that come from Lloyd's index of mean crowding, but it's a different equation. And so this is Lloyd's index of mean crowding. The statistic for mean crowding is equal to the mean plus the variance divided by the mean minus 1, okay? And you go out and you sample, and you calculate the means and variances for each of your samples, and then that gives you values. So Iweo took that, and he came up with this equation, alpha plus beta times the mean, where M is mean density, alpha is the index of contagion, and B is a measure of aggregation. Okay, so it will tell you whether or not it's clumped or not clumped. Taylor took it. This is the Taylor's power law that Ekbom used. The variance is equal to the scaling factor, and the mean brought to the level of the slope. And then he said, take a log regression of that, and it'll give you the values that you need. So step 1, density information. I'm not sure that we could do this today. Literally, we sampled 17 eggplant fields twice weekly during the growing season from 1989 to 1991. Cost a lot of money. I think we had a crew of 8 to 10 students helping us do that every
We
summer.
couldn't do that today because we can't afford the students.
Right, exactly.
Okay.
I can't remember what the minimum wage was, but it wasn't $15 a year. And so we went out and we did exactly what the scouts with the Department Ag did. we flagged 100 plants, and we went every week and sampled those 100 plants. Recorded the number of egg masses. We grouped larvae into small larvae, which were first and second instars, and large larvae, which were thirds and fourths, and of course the adults. We then standardized the small larvae and adult counts, and then calculated our means and standard, the variances for and we also did egg masses, which was very important early in the season. So step 2, use Taylor's power law, do the slope so that we could get the regressions to get the slopes. We needed to test for homogeneity between years, and if the slopes were not significantly different, we would have been able to combine all that data. They weren't significantly different, so we dealt with it, but we had to do it per year. And so this is the results of that. And the important thing here is, first, everything is a highly significant relationship with the R-squares here, which was good. And the second important thing here is that all of the slopes were above 1, which indicates aggregated distribution. Okay, and so that was good for us. When we compared the regression slopes between years, we did find that there was a significant difference in the slopes. Okay, and same thing for the combined larvae. So this was a t-test that we did with this, and again, it describes an aggregated distribution, which, if you've ever worked with Colorado potato beetle, it makes sense. You can see it very clearly in the field. It's not quite as clear as it is with aphids, but you can see it in the field. Okay, the next step, optimum number of samples. So, the whole point of what we're trying to do is come up with a program that means scouts can go out and do less sampling. Okay, and so you do that using this equation, where the number of samples is equal to 1 divided by your precision level. And so we chose a precision level of 0.25, and that has to do with, what do you want your possibility of making a wrong decision when it's a right decision, and the reverse. The value of A from Taylor's power law, and then the mean density. Okay, and so this is what you get. Okay, you get these curves, and we have individuals per plant at the bottom versus the number of samples needed to estimate density. And if you notice there, I've got a red line across both of those. At low densities of egg masses, which is what we chose to use as our base, that's 25 samples per sampling event. And so we use that for both egg masses, and we use that for the larvae and the adults. And so what that means is we at minimum need to go and do 25 samples versus the 100 samples that were being done. Okay, of course, now you never know how accurate your estimates are going to be. And so we developed all of this, and we submitted it for publication, and the reviewer said, well, you need to do some validation. And it turned out there was a computer program out there. It came, we got it from the people that wrote it. It came on a 2.5 inch floppy disk, the little tiny ones. How many of you remember those? Yeah, I don't have a machine that'll run the program anymore. Well, I got a machine, but I don't have a disk drive. So it's called resampling validation. We used a precision level of 0.25, and from all the data we had, we picked 2 fields to do this with, and the reviewer said run the software, and if it's in agreement this is working right, then we'll publish the paper. And so this is the output from that, and it's like any other random sampling. You would pick a mean from the data on a date and go through and run this thing, and it, on the old PCs, it's kind of like what the DNA analysis was on a Mac. The thing took hours. and it wasn't nearly as complicated as doing DNA analysis. And so I just want to point out here a couple of examples. And so from the sampling that we had estimates on June 30th of 1.4 individuals per plant and 1.09. And the mean number of plants by the simulation were very, very similar. And so there are several, that's at relatively high densities in this, then medium, and you see a similar thing, and then with low. And then here's the number of mean samples. Notice that none of those mean samples that the simulation said we needed to take was 100 samples. Okay, so that was good indication that we had something here, and now it's just a matter of putting it together. And so this is for the larvae and adults, it's the same kind of thing. Yep, I'm going to run out of time. Okay, and the number of samples needed. And so what? How do we use this? Well, this is what we call sampling lines, okay? And you can do this as a table, or you can do it as a graph, and the scout can take this, go out into the field, start their samples, and when they get to 25 samples, very quickly with a hand calculator, not on your phone because we didn't have phones back there, determine the density very quickly and then compare it to the graph. And the way this works is, we follow the line over here at 25 samples and come up, and it says, okay, at this point it's about 100. Okay, if we didn't collect more than 100 individuals on the graph, that puts us into this. If we do, it puts us into the stop sampling. Okay, so 25 samples, we're done. If that number at 25 samples falls between the two lines, that's the no decision area, and it says go do more sampling. Okay, and you would sample, and then at this get up to here. If it was, this is probably about 275 to 80 individuals. That says stop. Go tell the grower to spray the field. Okay, and I'll be honest with you, it worked very well, and most of the time we stopped at 25. Okay, so just a couple of conclusions. Taylor's power law regression showed similar significant aggregated dispersion for both eggs and the combined larvae and adults. Optimal sample size, 25 plants per egg masses, and the validation found that it fit and it was working. So this program was used by the Department of Agriculture Scouts. At its peak, we had growers waiting in line to get into this program, and we had problems because there wasn't a limit as to the number of parasitoids that the lab could produce, and at one point they were up to producing 10,000 of them a week, and it still wasn't enough for all the demand out there. And then in 1995, the program stopped. Does anybody remember their insecticide history? Ann does. What happened in 1995? Imidacloprid came on the market. It was specifically developed to control Colorado potato beetle. That was the first thing it was labeled for. And that was because potato beetle had become resistant to all the organophosphates, carbamates, and most of the pyrethroids. Actually, the beetle was, both larvae and adults. And probably the best thing to kill them was rotenone. Who's heard of rotenone?
Oh, yeah.
Yeah, plant material, it's a botanical, very good against them. And in fact, when we went through, and remember I talked about, the thresholds, if with egg masses they got to a threshold there, we would tell them to spray, because there was a disconnect between the emergence of the adults, which is based on soil temperature, and the temperatures, air temperatures that the parasitoid could withstand. It was from South America. It wasn't adapted to our climate. It couldn't overwinter. And so we couldn't release it until it was a certain air temperature. And so we'd do the samples, and if it hit a threshold, we'd say spray rotenone, because that was the thing that was least toxic to the parasitoids. And but imidacloprid knocked it out. Dr. Lasham always told the growers that if something that's more cost effective comes along, that's fine, and we'll stop the program. And imidacloprid was much cheaper. All they had to do was put all the transplants on a tractor, pull it out of the greenhouse, do a drench, and they would get almost season-long control
How
from it.
long did it take for insecticide for resistance to imidacloprid?
Probably about 5-6 years. Yeah, at least demonstrated in the laboratory. The immediate effect of all of this, though, was basically it knocked down the background level of Colorado potato beetle, and they're no longer as big a problem as they used to be.
So the imidacloprid did
Yeah,
that?
imidacloprid did that. Yeah. Okay, last part of this is the profit loss analysis. So we went out and we compared the inputs and outputs between the conventional growers and the biocontrol BCIPM, biocontrol IPM program. In 1993 and 94, every year we went to the growers and conventional and others and got their acreage data. We collected spray information, which if you've ever tried to do that is very, very hard. Even when it's mandated by DEP to be supplied every 3 years. They have to keep records. We collected harvest data for this. Myself and other people would stand at the packing line and count the number ones, number twos, and large. Number ones are perfect fruit, and that's what they get the most money for. Twos are the next grade down, and large, they're okay, but they've got some damage. They got maybe 1 or 2 bites on it. We also included what the growers were paying per hectare to be in the program, and then fortunately, and without it, we wouldn't have been able to do it. Dylan and Latimer were 2 ag economists in the ag econ department here at Rutgers, and they had done a budget analysis of all the inputs and what it cost for growers to grow eggplant here in the state. And we took that, and all we had to do was add in the cost of the insecticides to do this. So this is the results, and I'll just point you down here, and big difference in the kilograms of active ingredient applied in 93 and in 94 between conventional and the biocontrol growers. And so it was immediately obvious they were spraying less, and if I didn't say it, they were spraying 18 times a year, and it went down to 2, and that 2 was for egg masses. It wasn't actually for the adults and larvae, because the parasitoids were killing everything off. Same thing in 93 with the number of applications, except in 93 we had more in terms of applications for the biocontrol growers. And if you look at that, the reason for that, well, I can't go up with it, so I'll do it this way. Right here, they did have some problems, and so I don't remember why that actually occurred. But you know, the other thing here is most of the time rotenone was what they were using. Okay. And then in 94, it went back to the way it was for the kilograms applied. There was less by the biocontrol growers. Applications per year, again, significant difference between mean number of applications between the biocontrol and conventional growers, with the biocontrol growers doing less. Same thing with the number of treated acres and the total cost per grower in terms of dollars. significant difference. And, you know, that is amazing when you consider, you know, by today's standards, these old materials were dirt cheap. They weren't at the time, but based on today, they were dirt cheap. And also cost per hectare. And then finally, if we look at the number of boxes per hectare, it's over, you know, we've got more in for the number ones in terms of the biocontrol growers. We've got more in the number twos. And we've got, in 93, a mean of 0 doesn't happen very often. That basically means they didn't have any larges because there was no damage in that. And we had very low damage in 94. Number of boxes per hectare, no significant difference there, but the value, there were significant differences there with the biocontrol growers getting better return for what they were harvesting. In terms of costs, similar story, except in 93. But if you look at the insecticides, I think that was hand labor picking, because they had more fruit to pick from the plants.
George, does this include the price of making the parasitoids, or the parasitoids being provided by USDA?
Well, it was New Jersey Department of Ag. That's okay. So no, that charge, we say it was a charge for parasitoids, but it was actually that's what was used to pay the scouts. So now, if we included, I know where you're going, if we included the cost of producing the parasitoids, this probably would not have been cost effective. But we didn't look at it, so I can't say for sure, but my guess would be, which is kind of you know, yeah.
I'm all for the parasitoids, don't get me
Right,
wrong.
oh no, I know, yeah. And we look at this in terms of conventional, the profit and the crop value, and there are big differences here in both years. Okay, so conclusions. Biocontrol IPM growers reduced their pesticide use, they had higher yields, and they had higher net profits. I mentioned that we stopped doing this program. There were other places here in the U.S. that were rearing adobo. Maryland was doing it, and the USDA rearing facility in Texas was doing it. And there were labs in Italy, because on the consumption paper, Apara was actually a visiting scientist from Italy, and he took this back and convinced his government to build a lab just to rear these things, because their eggplant production is bigger than ours is. But even nobody rears this insect anymore. So we'd have to go back to, if I remember, it was mountains in Chile, and recollect this, because it's not around anymore. All right, so I've been way too long-winded, but I've got 5 of, so.
I've got 10 of
10 of? Okay. Wow.
9 of.
So how many of you are familiar with this insect? Yeah, okay. So this is the one that kind of changed the life of my lab. Brought in lots and lots of money to do lots and lots of work. Not initially, but eventually it did. And this is the insect that Dr. Nielsen worked with for her PhD, Heliomorpha hales. And it was native to Asia. It was introduced into Allentown, Pennsylvania. We're not sure how. We think it came in shipping coming out of the ports, because it's a shipping hub in Allentown in the mid to late 1990s. Our first find of it was in a blacklight trap in '99 that was being run in Hunterdon County by our vegetable IPM program. I said it's native to Asia. It is native to Japan, Korea, Taiwan, Myanmar, and China. Okay, and it very well, within 10 years it spread to most states here in the US, and also a couple of provinces in Canada. And so the only ones here that it didn't in the U.S. are Wyoming and South Dakota. Okay? And the other ones, I could say, I guess, because they only grow grain, and it doesn't really feed on grain. But I could say that for other states. It just never made it to the big commercial centers, which is how it got into a lot of these states. This is a map that was produced on a yearly basis by all of us. We're in the brown marmorated stink bug working group, and distributed through stopbmsb.org, and we ranked them as to whether they were a nuisance, an ag problem, or both. Okay, Canada is a little more widespread. It's made it into British Columbia, it's made it into Alberta, and it's in Ontario and Quebec. Okay, that's its origin, and so where is it now? Well, it's all over North America, it's all over Europe, it's all over New Zealand, it's in Australia, and it's also in Santiago in Chile. So it's now worldwide in distribution. And a lot of the thinking about it is some of this actually came from the U.S. because they were shipping things out of country that had them in the packaging. And it was a concern of several countries, New Zealand the most, where they actually required cars coming to them from an known infested area that they had to either be heat treated or sprayed before they could get off the boat. So, serious problem. This is courtesy of Dr. Nielsen. It's one of her slides. They go through 1 to 2 generations, maybe 3. In the fall, the adults, of course, go to structures, and they feed on. They don't feed, but people don't like them when you get thousands of them, in their house. They overwinter there. They then go back out, and they go and they feed and go through those 2 generations. They have 5 instars, and those red, orange, and black ones on your left are the first instars. Okay, they feed on a wide variety of crops. We used to laugh, pick a crop and it probably feeds on it, okay? And so I've just got a small smattering here of not only agricultural pests, but ornamentals. They actually are able to put their stylets through the bark of thin-walled trees and pull out fluids to feed on. So they feed on a lot of different things. We were going on, and Ann and I were kind of the only people working on this, and some other people. It was starting to become a problem in Maryland and a couple of other West Virginia. And in 2010 was the big outbreak in the East, and with the populations exploded, and bed bugs were a big thing. And basically, this says move over bed bugs. BMSB is now a bigger problem than bed bugs were. And so now what? And so what you're seeing here is the stink bug feeding on soybeans. Well, we went to the literature, and there wasn't much there, at least initially. And then later on, USDA group in Kearneysville, West Virginia, were able to get at the Asian literature and have it translated. And it turned out there was a lot more there than what we thought. But we had a lot of questions. One, where did it come from? And Ann was interested in this, and she convinced Dr. Fonseca to become interested in this, and so we did some work there. We didn't know anything about its development rate, so Dr. Nielsen worked all of that out. Seasonality and what crops and what plants it fed on needed to be worked out. And then the big question, because I'm in extension, and the extension folks were now worried, is how do we deal with this? Okay, because we didn't have a lot to do with that. And so I'm going to talk about, I'll have time to talk very quickly about this. Where did it come from? Well, we were able to get samples from different parts of the United States, including the West Coast, because it was in the Portland area, and had gotten into California, although they wouldn't admit it, for a number of years. Japan, Korea, and we got a number of Chinese samples, thanks to Kim Holmer, who is a biocontrol person with the USDA Biocontrol Lab in Newark, Delaware, who was doing research over there looking for parasitoids of another, and he sent us samples. So Dr. Fonseca's lab did the work. They sequenced the mitochondrial genes. We looked at CO1 and CO2 and 12S ribosomal DNA, and then conducted a haplotype analysis. And so this is just a pictorial of the different haplotypes from the samples in Asia. And if you look at this, there's basically 4 different haplotypes and different combinations. And then we looked at the haplotypes from the US samples, and we found 2. And the ultimate decision was that the closest alignment with Asia and here, the samples came where they came from Beijing. Okay. Now that has changed a little bit because we have more samples. Yeah, but that's what we were able to determine. Development rate. So this was done at a number of different temperatures. Didn't we used to have 5 degrees in there, but nothing hatched? Or was it?
Nothing hatched at 15.
Nothing hatched at 15, okay. And so what you do with this, you do different temperatures, rear them, and you figure out, the minimum development temperature and the maximum. Dr. Nielsen used 3 different models to look at this and compare which one was the best one to use, and then it was used to calculate degree days. And Dr. Nielsen has made full advantage of this and actually has developed models with other people so that if we know the temperatures, we can calculate degree days to certain points anywhere here pretty much in the United States to figure out when we need to do stuff. This is a standard graph. The one that most people, the simple one everybody does is this linear one. And with her work, the lactin actually fit the model, and you can see it's a curvilinear model. And so we use 14.2 as the minimum, and 35.8 as the maximum. And she was able to calculate that they go through 538 degree days throughout their life, and that females need 148 days pre-oviposition before they start laying eggs. And that's what she made use of that. Okay, seasonality. I'm out of time. Okay, I just want to go, I'm going to go real quickly through this. I'm just going to say, what did we do for the growers? We asked our vegetable IPM program to start pulling these out of their black lights because they were attracted to it, and they had 70 to 90 of these traps all over the state depending on year. And based on their counts from those, Dr. Nielsen was able to look at the spread through the state and then actually came up with a model to predict the rate of invasion and so forth. And with that, if you want to see the rest of it, make it a meeting with me and I'll show you the rest of it. Are there any questions? Yeah.
So how did they choose you to be the first person in the U.S. to work on brown Maramorosch extinction?
Very interesting story. So Allentown, Pennsylvania is the base of the office for the Lehigh County Cooperative Extension in Pennsylvania. And they had a equivalent of a program associate. I don't know, Karen Bernhardt. She had an entomology background. She had a master's degree, and I forget where she got it from, but I think it was one of the Midwestern departments. She had gotten calls because they were having the big problems with the stink bugs coming into people's houses in the late fall and congregating in large numbers on plant material on their landscapes. And initially, they thought it was the brown stink bug. But then at some point she realized that doesn't really look like a brown one, and she sent it to Cornell, to Rich Hobeck, who was a certified identifier for aphis, and he said this is brown marmorated stink bug. We were closer, I think, than Penn State, and so she called me and Dr. Peter Shearer, who used to be in our department, and who Dr. Nielsen replaced when he left to go to Oregon. And we went out there and we took a look at it, and Dr. Shearer had done his PhD with stink bugs that attack macadamia nuts in Hawaii. Talk about a great place to do research. And he said, I've never seen so many stink bugs in my life in one place. So it was classic, invasion theory, where you have this huge expansion. And then we were seeing the beginning of that. And we kind of agreed that we would start working on it at that point. There was also a person from APHIS, Gary Bernan, who was interested in this, and he was helping getting this going. Unfortunately, he couldn't get APHIS interested in doing anything. And so we started doing Ann's work. She also did work with pheromone traps. Most of it in the beginning was done, all except for the temperature work, done in Allentown, Pennsylvania, over next to Gorney Park.
You also had already those black light traps. So you
Yeah, they were there.
Yeah, and so
Yeah.
then you were starting
Yeah.
to see that. I remember you coming to talk to me and showing me these data where you have,
Right.
oh, this is when we saw them in 1999, and then just, so, yeah, that
Yeah.
is so
Yep.
cool.
And
I
that,
don't think I
So-
started
that-
going through them until 2006.
right, right.
We just happened to have all of them.
We had them all there, yeah. And so they've done this for years, and they make these geo-reference maps for corn earworm and corn borer, and put these out on the web for the growers to look at. And we started doing that with this. And this is just kind of a slide of the
Oh, boy.
massive
Yeah,
outbreak.
having it over time is a little bit like museums, where you
Yeah.
go back and like, oh,
Right,
look,
right.
this stuff was there, nobody noticed.
And so that's another one.
Yeah.
Yeah.
And I think that's what you showed me, like right there
Yep.
came from Pennsylvania, Allentown, just right across the river.
And so that's how we got started. And then it hit West Virginia and parts of Maryland, and explosions. And in the year that it blew out in 2010, tree fruit growers lost their whole crop in a lot of situations because they didn't realize it was there. Nobody did. And so they went to government, screamed bloody murder, and all of a sudden there was a grant program that allowed us
Yeah, but
to
it's
apply
always like
for it.
5 to 10 years after it's something should
Yes,
have been done.
yeah, yeah. Well, if APHIS had said this was nothing more than a homeowner nuisance problem in the 1990s, there would have been a lot more done
He
earlier.
was asked for my entire dissertation when I was done.
Yeah, no, yep,
They said whoopsie.
yep. They actually admitted it. They admitted it in one of the working group meetings.
Well, it's the same
Yeah.
thing with spotted lanternfly. I mean,
Yeah.
something
No,
that's
they had an
exploding.
immediate response.
Yeah.
Immediately it took 4 hours for 4 years for in Pennsylvania for anything to happen. Remember we went and visited in 2000?
No, but they already had money, it was just the money was.
Yeah,
more
it
for
it started
extension
with the
and
Department of
information.
Ag. Yeah, they were the first ones that they did the first presentation about it at an Eastern Branch meeting, and
You
everybody.
mean Eric's Fischiger at
Yeah.
Yeah, but he was crying bloody murder that nobody was paying attention to him.
Yeah, but it was a faster response.
Yeah.
It's all relative.
Any other questions?
I have questions.
Okay.
Yeah, I have questions about the entire thing about the aggregation with the Colorado potato beetle. So you mentioned that later. So the parasitoid came from Peru
Mhm.
because potatoes are from Peru.
Right.
And so you had to go back.
But I think it came from Chile.
Sorry.
Yeah.
Forgive me, Peru and Chile.
That's okay.
So it came from Chile. So so there's no parasitoid around here. So they always had to be the ones
Yeah.
from the original
Yeah.
evolutionary
Yeah,
origin.
and it was actually collected in Chile on a different species of Leptinotarsa.
Ah.
It wasn't actually Colorado potato
Oh,
beetle.
that's interesting.
Yeah. The big problem was it wasn't able to diapause here. And so you had to do a whole new inundative release program every year.
Which means that there may be a parasitoid in actually Peru, which is high enough that
What's-
it may have
yeah.
diapausing
So
parasitoids.
the story with that is Ben Putler, i.e. Putleri.
Okay.
He was a USDA biocontrol person in their Columbia, Missouri lab, and he did his literature review. And the story goes, he got on a plane with no money. I don't know how he got on the plane. He went there.
Hitchhike.
He went.
It's
He had.
Ben, they just let him on.
Yeah,
Just
I
talk
think
their ear
so.
off.
I think so. And he was a character, but a good entomologist. Got off the plane. He had a couple of locations he wanted to go, and he found it. And
But so you had already, because you did a literature review.
yeah, and he got it back in the U.S. Back then,
Which you couldn't do now at
no,
all.
you couldn't. Well, you probably couldn't take it out of the country now.
Right.
Yeah, and he brought it back. He started a colony. They worked with this insect first in potatoes, and then it moved over. It wasn't as effective in potatoes as it was in eggplant, and I don't know if Jim would.
Yeah, that was my other question.
I don't know. I don't know if Jim would agree with me, but I've always thought it was the plant architecture, because potatoes is more of a prostrate.
Right.
I gotta not say prostate. It's
No,
prostrate plant, as opposed to eggplant. You know, it's more upright, which was more similar to the type of plant it was originally collected from.
Ah.
Yeah, so it was evolved for that.
So that was my, actually, sorry, you know me, I'll just keep on going. But I mean, the potato, so the potato growers, is the Colorado potato beetle a problem to potato growers? Yes, right?
Yes.
So, but they couldn't use, even the parasitoid did not work for them.
They weren't able to make it work successfully. Yeah. Plus, at the time Jim started working with this, potato grow acreage in the state was declining. So all you folks have driven down 130 down the Heights town, you know where all those beautiful warehouses are? That all used to be potato. Most of it used to be potato acreage. It's some of the best soil in the state for potatoes. All right.
Yeah, Bill, I think Heather.
Yeah, I
Yeah.
have a question. So if the Colorado potato beetle feeds on solanaceous plants, I feel like I would expect it to already be somewhat more resistant to nicotine,
Insecticides.
like specifically nicotine, right? Because that's derived from solanaceous plants.
Right.
So, like what?
Nicotinoids, not
Yeah,
necessarily
yeah,
nicotinoids.
like nicotine.
It's different.
So did they not?
So
see-
Colorado potato beetle is the poster child for insect resistance.
So what do they
It
use?
has
it's not.
become. It has in agriculture. it
Oh,
is.
in agriculture,
Yeah.
fine.
It's become resistant to everything pretty much they've thrown at it.
So what do they use now?
Neonicotinoids.
No potatoes.
Yeah, yeah.
They use dynamites?
They don't. I haven't worked with it in years because it became less of a problem. So I honestly don't really know. I'm guessing, but there are some. As Ann pointed out, there's some new products that are. At one point, they also used Bacillus thuringiensis. There was a strain Tenebrionis that was effective against the larvae. Growers didn't like that in the beginning because the larvae, when the insect feeds on it, whether it's a gypsy moth, spongy moth, or Colorado potato beetle, it takes a while to kill them, and they just kind of sit there in a stupor. And growers at that point wanted the bugs to fall off the plants because that's how they knew it was working. So Cesar is frowning.
Yeah, he's like with beetles. Usually they don't work as well. Like that's my
Yeah,
experience.
but it did work very well. Yeah, if they got it out there, just like with spongy moth, you got it out early enough on the smaller stages, they were susceptible to it.
So, but in a post-insecticide world, I mean, you see biocontrol of these types of parasitoids coming back. I mean, it's getting warmer and insecticides are not working. Is it too expensive?
I would, well, if somebody was able to recollect it and start a colony, I've always said when the insecticides fail, they're gonna have to do that. or we are going to have to accept eggplant that has little bites from the
Yeah,
adults.
which is
in it.
kind of silly because.
Yeah.
George, what about the soil building stage? Like, what about tillage or cover cropping or something?
Okay,
Or reflective
so
mulches? Like,
how many of you have heard of a compound called temic or aldicarb? Yeah, very toxic material, okay? They could use it as a soil application at planting, and it would knock the Colorado potato beetle dead. But it has this problem. It's very water soluble. And so, what kind of aquifers do we have down in South Jersey? In the other big area, what are they like out on Long Island? Because there used to be a huge potato growing region. It had the potential to get down into groundwater, and in some cases it did. And before it was canceled, we actually had labeling on it that said you couldn't use it on Long Island, and you couldn't use it in certain counties here in New Jersey. All right,
So
we're,
back to-
yeah, we've got to meet with the
Yeah.
class.
Yeah.
I know they're getting hungry.
Pizza's already waiting.
Well, thank you.
By the way, George, there was a lot of, really well from the online, but
Okay.
I didn't see any questions.
Chris, Diamond,
Hooray for IPA.
I'm looking to see you.
I guess if you're going after this,
I think that's actually what it says in the publication.
Okay.
Shut her down.
This is supposed to be recording, but I'm wondering if it is. I didn't stop
It
sharing.
was saying.
It was saying, okay. Thank you, everyone.